So, good morning, everyone. Welcome to the 2024 Symposium of the NIH Distinguished Scholars Program. I'm Karo Hashimoto. I'm Director of Faculty Development in the Office of Intramural Research, and I direct the DSP. Anyway, it's my great pleasure to introduce Dr. Nina Shore, who will give opening remarks to the NIH Research Festival as well as the Symposium. Nina.
Thank you, Dr. Hashimoto, and thanks to all of you for coming and for anyone who is listening to us online. As Dr. Hashimoto has said, I'm Nina Shore. I'm the NIH Deputy Director for Intramural Research, and I have the truly honor and pleasure every single year of opening the NIH Research Festival. I welcome all of you, both online and in person, to what I truly know will be 3 fantastic days of NIH science. The NIH Research Festival began in 1986 as NIH Research Day, and the festival has been held annually since then, with the exception of 1987, when they decided to celebrate the centennial of NIH instead. And in 2000, as you likely all know, the festival went virtual and presented the same cutting-edge science, but in a digital-only format. Last year, it came back in person with a virtual option for many of its offerings, and it was so successful that we decided to do much of the same thing this year, but with the hope that more and more of our audiences will be right there in the same room with the speakers. In addition to our symposia, workshops, poster sessions, lectures, and receptions, there are resource tables that I hope you will frequent during the festival. And new this year are the inclusion in the festival on Wednesday of the Dr. Philip Chen Jr. Lecture on Innovation and Technology Transfer, the Victoria A. Hardin Lecture on NIH History, and this week's Walls Lectures. What a lineup we have. Well, the Research Festival showcases the breadth and depth of NIH intramural science, the diversity of viewpoints, approaches, and scientific backgrounds of the NIH intramural scientists, and the joy and challenge of finding answers to questions that no one else even thought to ask. I hope that over the next 3 days, every NIH intramural scientist will get to take part in some way in the presentations, the festivities, and the collaborative spirit of the NIH Research Festival. So it's no accident that the Distinguished Scholars Program Symposium is the opening event of the 2024 NIH Research Festival. Indeed, dedication of the DSP to welcoming, nurturing, and supporting a scientific workforce that represents the full breadth both of humankind and of intellectual pursuit mirrors precisely what NIH sees as the best future for science and medicine. The symposium will showcase the work of intramural investigators at different stages of their careers, engaged in studies of different kinds of biomedical and behavioral science. Together, they continue to make each other wiser, more creative, and more questioning of the scientific status quo. So it's truly a pleasure to open the Distinguished Scholars Program Symposium and to approach the day when anyone who aspires to and works towards a career in science or medicine can honestly say, I belong. And now I'm going to turn the podium over to our distinguished scholar, Master of Ceremonies.
Thank you. Thank you very much, Dr. Shore, Dr. Hashimoto, and the DSP for hosting this. This is especially, we were just having a conversation before the start that meeting in person is so much more conducive to innovation, and so it's really nice to see many of you in the audience here and those of you online as well. So for our first speaker is Dr. Angela Ballesteros from NIDCD, who's going to be presenting to us on structural and functional insights into the mechanical electrical transduction channel essential for hearing.
So hi everybody, and thank you so much for joining us. Please allow me to start this amazing DSP symposium by giving you some structural and functional insights into the mechanical electrical transduction channel essential for hearing. So when I started my postdoc, I became very interested in contributing to answering the question that is shown here in pink. And is that even if we knew the molecules that were responsible for sensing the other senses, we didn't know the molecular identity of the mechanoelectrical transduction channel responsible for hearing and balance. And I was very shocked by this because hearing loss is the most common sensory disorder, and it actually affects to a lot of people. And those numbers are dramatically increasing now that we live longer and we move to industrialized cities where we are exposed to a lot of noise. And I'm very happy and proud to be able to say that the work that has been performed by a lot of different labs and my own work, we can now say that TMC1 and TMC2 are the mechanoelectrical transduction channel responsible for sensing hearing and balance. So let me tell you a little bit about the system that we work with. So the sound waves had to travel all the way from the outer ear through the middle ear into the inner ear, where we have this spiral-shaped organ that is called the cochlea. And inside the cochlea is where we have the auditory sensory organ that is called the organ of Corti. And in a very specific region, all along this organ is where we have the auditory sensory cells that we call them the hair cells. And they are called the hair cells of the inner ear because they have those hair-like filaments that we call stereocilia, that they are organized in bundles. So every hair cell has one bundle of stereocilia that is located at the apical region of the sensory hair cells, kind of like resembling a mohawk. And this stereocilia bundle is the mechanosensory organelle of the sensory hair cell that is responsible of sensing the mechanical stimuli that is coming from the sound and transforming these mechanical stimuli into electrical signals that are going to then be transferred to the brain. So let me tell you a little bit about how the mechanosensory organelle of the hair cell, the stereocilia bundle, does this. So we have a protein filament that is called the tippling that connects the plasma membrane of the longest stereocilia with the tip of the shorter stereocilia. And when the sound waves go into our inner ear and they bend and the stereocilia bundle towards the longest stereocilia, the tippling is going to pull from the membrane of the shorter stereocilia row, leading to the opening of this mechanosensitive ionic channel that is then going to allow for the permeation of those positively charged ions into the hair cells. So we knew of at least 4 different transmembrane proteins that they were thought to interact with each other, forming that we call the mechanoelectrical transduction complex. And all those proteins are essential for hearing, but one of the big questions that remain open in the field was which one of these transmembrane proteins was actually the ion channel and have was the pore-forming subunit of the mechanoelectrical transduction complex. So knowing the identity of these proteins was not only important to identify the molecule responsible for sensing hearing and balance, but also because we have a lot of ototoxic drugs that they are commonly used in clinic, like, for example, the aminoglycosides antibiotics and the anticancerogen drug cisplatin, that they can also permeate through this channel and they accumulate into our hair cells and they actually kill the hair cells, leading to permanent hearing loss. So by identifying this molecule and getting some structural and functional information in this molecule, we can also motivate drug design studies that will reduce the ototoxicity of these molecules. So we have a lot of experimental evidence pointing out to the TMC proteins, so here in gray, as being this ionic channel. But one of the things that we did was to try to generate a structural model of these proteins so we could get some structural information and see how these proteins look like. So what we did was to look for a distant related protein that we already have a structure that we could use as a template to model the structure of TMC1. And when we did that, we found out that there are two other family of proteins, the OSCA or TMEM63 and the TMEM16 proteins, that they have a lot of conserved regions with the TMC proteins. And these two other family of proteins, they are actually paired with the TMCs of the same superfamily of proteins, and it was shown in this evolutionary study. So we have more information about these two other proteins. We have structural information. We know how those proteins look like, and we also know what is their function. So the TMEM63 and the TMEM16 proteins, they both families of these proteins can function as anionic channels, which already suggests that the TMCs may also function as a channel. But very interestingly is that the TMEM16 proteins, they can have also this membrane regulatory lipid scramblase function that I will go back in a couple of slides. So this is how the structural model of TMC looks like. This is the first structural information that we got on the TMC proteins. They are dimer, they have 10 transmembrane domains. More importantly, the generation of this structural model allows us to locate 22 different TMC1 deafness causing mutations. So this is going to help us understand how these point mutations alter the function of these proteins and lead to hearing loss. When we look from the side, we can see the presence of a cavity at the protein member interface, and this cavity functions as the ion permeation pathway or the pore in the related TMEM63 and the TMEM16 proteins, and we thought that it could also function as a pore in the TMC proteins, as the pore of the mechanoelectrical transduction channel. So we look at this pore a little bit more in detail. So I'm going to show you now a picture looking from the sides when we can actually see this cavity in surface representation. And this arrow highlights the permeation pathway that those ions or the autotoxic drugs will have to follow in order to enter the hair cells. So this structural model not only supports a lot of functional data that we have from electrophysiology experiments performed in hair cells from different vertebrate models, but it also supports the role of the TMC proteins as the mechanoelectrical transduction channel essential for hearing and balance. But one of the cool things, or the things that I kept wondering when I look at this structural model, was that this pore is partially open to the plasma membrane. So I kept wondering how the positively charged ions are sealed from the hydrophobic core of the plasma membrane. And I look at how those TMEM16 proteins, that they can not only function as ionic channel, but also a lipid scramblase, work. So in the TMEM16 proteins, that they have this dual function, the lipid scramblase function, they allows for the permeation of phospholipids from one side of the plasma membrane to the other one. And the phospholipid head group, represented here in red, is thought to participate in the sealing of those positively charged ions, of these charged ions from the hydrophobic core of the plasma membrane. So I hypothesized that maybe the TMC proteins can not only function as anionic channels, but maybe they can also have this lipid scramblase function and somehow regulate the hair cell membrane. So before I show you a little bit of data, I need to tell you about how those lipid scramblases work. So the plasma membrane, the cell membrane, the plasma membrane is asymmetric, meaning that we have specific phospholipids, like phosphatidylserine, that they are always located in the intracellular side. They are looking at the inside of the cells. And when we have an activated lipid scramblase that is present in the cellular membrane, what happens is that this protein allows for the transport of phospholipids, and it leads to the homogenization of the plasma membrane and the externalization of these phospholipids that they are usually not accessible. We also lost a lot of interactions between the plasma membrane and the cellular cytoskeleton, so we can observe the presence of those membrane blebs that are being generated in the plasma membrane. We can use a probe that is fluorescently labeled to detect those phospholipids, and by doing these kind of assays, we can not only test the potential existence of a lipid scramblase at the hair cell bundle, but also to test the hypothesis of the TMCs playing a role in this new function. So I don't have time to show you here all the different experiments that we perform using knockout models for the TMC proteins, different manipulations, and even organocorti explants from mouse models carrying point mutations in TMC1. But what we saw is that there is this TMC1-dependent phosphatidylserine externalization and membrane blebing that is happening at the stereocilia bundle, and suggests that the TMC proteins can actually also function as lipid scramblase and regulate the hair cell membrane. So what we think that is happening is that when the ion channel function of the TMC proteins is working, the plasma membrane asymmetry is preserved. But when we have deafness-causing mutations in TMC1 or a decrease of the intracellular calcium concentration, this lipid scramblase function gets activated, the phosphatidylserine gets externalized, we observe the presence of these membrane blebs that they are actually not only alter the properties of the plasma membrane, but they also mislocalize some of the proteins that are essential for mechaноtransduction. And we think that when this mechanism is happening in a regulated way, it may be a mechanism that the hair cells are using to recycle non-functional mechaноtransduction channel complex. But when it's happening in an irregulated way, it may lead to hair cell death and hearing loss. And our lab is not only interested in the characterization of the structural and functional characterization of the TMC proteins, but also in understanding how this membrane regulatory function alters hair cell physiology. So with that, I would like to thank my former lab, my mentors, my collaborators for all the support, the DSP program for this amazing opportunity, and I couldn't be more excited to introduce you to the new section of sensory physiology and biophysics at the National Institute of Deafness and Other Communication Disorders. And with this, I will be happy to take any questions if I have time.
Thank you, Dr. Ballesteros. If anyone has questions, please come up to the mic, introduce yourselves, and pose your question. If there are any questions online, do we have someone monitoring in the back? Wonderful. Well, this is great because I have a lot of questions. So fantastic. So by background, I'm a vascular cardiologist, and so this is very interesting to me. We think of these TMEM16 proteins, these scramblases, as being very important in regulating endothelial health because these phospholipids get exposed, and then we start to get lots and lots of clot formation. So I'm just thinking about TMEMs and wondering if the scramblases and the flipases, if you notice that the flipases have a similar phenotype that's modulated by TMC1, or is this restricted really to the scramblase family?
That's a really good question. So we know that mutations on flipases and flopases, they also cause hearing loss and deafness. So definitely membrane regulation is very important for proper hearing and balance. But for now, we think, and this is a new thing, we are still working, we need to do more work. We think that the TMCs function as lipid scramblases because based on the preliminary data that we are generating now in our lab, we know that they are not lipid-specific like other lipid scramblases. And so that suggests that, and they don't need energy like the flipases and the flopases, so they're a little bit different. And they are also not regulated as the TMEM16. So they have some things in common with other lipid scramblases and lipid flipases and flopases, but they are different in some ways.
That's wonderful. Thank you. Do the hair cells regenerate?
They do not.
So once you
forgot to mention that. Thank you.
So if they don't regenerate, I think then there's an importance to preserving them.
Exactly.
Interesting.
Yeah. Thank you so much.
Are there other questions from the audience? Okay, wonderful. Well, thank you very much, Dr. Ballesteros. So I try to take every seminar and turn it into cardiovascular. So our next speaker is Dr. Jacqueline Vo, who is from NCI, and will be speaking with us about treatment-related cardiovascular disease and health inequities among breast cancer survivors. And Dr. Vo is going to be presenting to us virtually. Welcome, Dr. Vo. Dr. Vo, you may need to unmute your mic.
Can you hear me now?
Yes, beautifully. Thank you.
Okay. Great. Sorry about that. Thank you, Dr. Gansey, for the introduction, and good morning from Los Angeles, California. Thank you to Dr. Hashimoto and Isanaka for the coordination and flexibility to facilitate my remote presentation as I am at the AACR Cancer Health Disparities Conference this week. I am part of the 2023 NIH Distinguished Scholar Program, and I am a tenure track investigator at the NCI Division of Cancer Epidemiology and Genetics, and I'm delighted to share a brief description of my research portfolio. Next slide. There are over 4 million breast cancer survivors in the United States, comprising over 20% of the cancer survivor population. Breast cancer survivors are living much longer due to advances in screening and treatment, contributing to a 91% 5-year relative survival. But just because they are living longer doesn't necessarily mean that they're living better. Many of these cancer treatments that have improved survival can unfortunately cause adverse health effects. Next slide. Breast cancer survivors are at increased risk of cardiovascular disease compared to women without breast cancer, and this is in part due to cardiotoxic cancer treatments such as anthracycline-based chemotherapy or trastuzumab, as well as shared risk factors between cardiovascular disease and breast cancer. Next slide. Using SEER data, we've reported an increasing trend in heart disease mortality among women diagnosed with regional stage breast cancer who were treated with chemotherapy from 1975 to 2016. We hypothesized that this risk is driven by anthracycline-based chemotherapy, but unfortunately, SEER does not have specific chemotherapy types. Next slide. Current clinical guidelines were developed to recommend cardiac surveillance among patients who receive cardiotoxic cancer treatment up to 2 years after treatment completion. Yet clinical guidelines have reported that they are largely based on clinical experience due to the lack of data-driven studies, and they currently do not have long-term or age-specific recommendations. Next slide. I'm going to present the findings of one of my studies, and the present study aim was to evaluate the long-term risk of incident cardiovascular disease after anthracycline and trastuzumab treatments among approximately 10,000 one-year breast cancer survivors. The detailed exclusions are described to the flow diagram to the right. Next slide. So to conduct the study, we leveraged the Kaiser Permanente breast cancer survivors cohort, which is a long-standing retrospective cohort with prospective follow-up using data from cancer registries, electronic medical records, pharmacy files, and death data. And the co-PI of this cohort, alongside Dr. Gretchen Gerack, and our NCI DCEG team is highlighted to the right, as well as our KP collaborators right below. The present study leveraged data from Kaiser Permanente Washington and Colorado, but we are in the works of expanding to Kaiser Permanente Georgia and Hawaii. Next slide. Our primary exposure of interest is chemotherapy regimen type based on risk of cardiotoxicity, including anthracycline and or trastuzumab, other chemotherapies, and no chemotherapy, which is our reference group. For our outcome of interest, we included cause-specific cardiovascular disease, including incident cardiomyopathy and heart failure, ischemic heart disease, and stroke. For the sake of time, I will present only the incident cardiomyopathy and heart failure results. Next slide. For our analysis, we conducted cumulative incidence accounting for competing events, and we then calculated hazard ratios using Cox proportional hazard regression models with attained age as the time scale to account for the naturally occurring increase in cardiovascular risk with age, and adjusted for patient characteristics, other breast cancer treatments, and cardiovascular disease risk factors. Next slide. These are the patient and breast cancer characteristics. Next. Oh, stay with this. And women treated with anthracyclines and or trastuzumab were diagnosed with younger ages, at more advanced stage, as well as a more aggressive form of breast cancer, including estrogen and progesterone receptor negative breast cancer, compared to women who received other chemotherapies or no chemotherapy. Next slide. These graphs show the cumulative incidence of cardiomyopathy and heart failure by treatment type, stratified by age at breast cancer diagnosis. So on the left graph includes women who are less than 55 years of age. The middle graph includes women 55 to 64 years of age. And the right graph includes women 65 years and older. On the x-axis is years since breast cancer diagnosis. Next. At 10 years after breast cancer diagnosis, next. The greatest absolute risk differences for cardiomyopathy and heart failure were among women who were less than 65 years, treated with anthracycline and/or trastuzumab compared to women who received other chemotherapies or no chemotherapy. You can see that the higher red line corresponding to the woman treated with anthracycline and/or trastuzumab. Specifically, the 10-year cumulative incidence was 1 in 29 for women aged less than 55 years and 1 in 10 for women aged 55 to 64 years. There were no clear differences by treatment for women older than 65. Next slide. We then assessed the relative associations for anthracycline and trastuzumab treatment on cause-specific cardiovascular disease risk compared to women who did not receive chemotherapy overall and stratified by years since breast cancer diagnosis and age at breast cancer diagnosis. The hazard ratios are on the x-axis. Next. Women who were treated with anthracyclines and/or trastuzumab had a higher risk of developing cardiomyopathy and heart failure compared to women who did not receive chemotherapy after adjusting for breast cancer characteristics or other treatments. Next. These risks persisted long-term from 5 to 9 years after breast cancer diagnosis, as well as 10 plus years after breast cancer diagnosis. We also found really striking results for breast cancer survivors who were aged less than 65 years. The highest risks were observed among the youngest group of women aged 20 to 54 years who received anthracyclines and/or trastuzumab. They had a 3 times higher risk of developing cardiomyopathy or heart failure compared to those who did not receive any chemotherapy. Next slide. So across 24 years of follow-up, we observed that breast cancer survivors treated with anthracyclines and/or trastuzumab were at increased risk of developing cardiomyopathy or heart failure, and these risks persisted long-term even after 10 years from breast cancer treatment. The greatest risks were among women who were diagnosed with breast cancer before age 65. Next slide. So not every patient who receives anthracycline or trastuzumab will go on to develop a cardiovascular disease. So who are the patients at highest risk for developing a treatment-related cardiovascular disease, and which high-risk patients should clinicians aim to intervene with? So our study implications were really to highlight the need to increase cardiac surveillance intervals for high-risk patients, especially the younger women who are surviving many decades after treatment ends. Next slide. Importantly, access to breast cancer treatments and survivorship care can also affect different populations unequally. I've conducted several descriptive epidemiology studies describing the disparities in increased cardiovascular disease mortality for Black, Latina, Asian American, and Pacific Islander breast cancer survivors, as well as women who are living in counties with the lowest socioeconomic status and greatest virality. Next slide. One area of health disparities research that I'm particularly interested in is in the disaggregation of Asian American and Pacific Islander populations who comprise different and distinct racial groups, yet unfortunately are aggregated in research. In this recent study that we've led using death certificate data, my team and I have described the leading causes of death separately for Asian American and Pacific Islander populations. And here I've highlighted the female population. Next slide. For both racial groups, cancer was the leading cause of death, yet cancer mortality was 2 times higher for Pacific Islander females compared to Asian American females. And this really highlights the importance of consistent disaggregation of these different racial groups so that we're not masking potential disparities for Pacific Islander populations who represent such a small percent of the total aggregated group. Next slide. So to accomplish my goal, I have led the expansion to increase the racial diversity within the Kaiser Permanente Breast Cancer Survivors cohort through the support of DCEG and the NIH Distinguished Scholar Program. By expanding to Kaiser Hawaii and Georgia, we were able to increase the proportion of Black women by 3 times, Asian American women by 4 times, and Pacific Islander women by 7 times in our cohort. Our team's next steps are to understand if and how cancer treatments may inequitably impact minoritized cancer survivors and to assess treatment-related adverse effects such as cardiovascular disease, second cancers, and mortality by race and ethnicity, as well as county-level socioeconomic status. Next slide. With that, I'd like to acknowledge and thank my mentors, my colleagues, and my collaborators, and my mentees in the NIH Distinguished Scholars Program. It's been a wonderful year of growth within the DSP, and I'm thankful to have the support of the program as well as the support of my DSP mentor, Dr. Paul Waite. I look forward to the years to come. Thank you.
Thank you very much, Dr. Vo. Leave it to the DSP scholars to stay on time. It's really quite interesting work, and congratulations on seeing some very direct, immediate outcomes of your work, where you're changing the way data is collected so that we can then think about what comes next after that. There are questions in the audience. Dr. Platt?
Yes, this is really nice, Jackie. Thank you for this. I'm curious about diabetes. It kept showing up, and we know that that's increased in certain minoritized populations. What is the suggestion, if any, for breast cancer doctors in dealing with patients with diabetes and some of the outcomes that you want to treat the breast cancer, but diabetes may be a complication? I'm curious, any advice on that?
That's a really great question and an important consideration, because we have to really, in these patient populations where the risk of comorbidities are really so common, like these diseases are often co-occurring. I don't have particular advice for clinicians at the moment for diabetes. It's an important area of research that really needs to be explored further. But as this diabetes was an important risk factor for cardiovascular disease, so in our study we did adjust for and account for diabetes, but it is something that we're considering in the future, is to consider how patients who present with multiple comorbidities, how their risk of cardiovascular disease may occur, particularly in relation to their cancer treatment. Thank you.
Hi, Jackie. This was amazing. Thank you. I was particularly intrigued by the fact that women that are older than 65 were kind of protected. So I was wondering if you had any hypotheses in terms of mechanism, whether that was because maybe they're in senescence already to some degree, and their heart cells are not as vulnerable to, the anthracycline treatment, and whether you plan on using any biomarker studies in the future to elucidate that. Thank you.
That's a really great question. I am cautious to say that among older women, that their heart would necessarily be protected from the cardiotoxicity. I think what we're really highlighting here is a potential difference in maybe healthcare and treatment access for the older patients, who we know that as you get older, your risk of cardiovascular disease increases. So generally, clinicians might be more cautious regarding whether or not they're going to give them an anthracycline cardiotoxic drug when they know that their risk of developing a cardiovascular disease is much higher than for a younger woman, for example, who are much healthier, have much less comorbidities. So I don't know if that's per se the consideration, but absolutely something we want to explore further, and absolutely biomarkers are a really important context. We've collected some information, but it's limited in the basis of the electronic medical records, but it's something that we're really trying to elucidate further in our next step. Thank you.
Thanks so much. That was really interesting work. You showed us your data on long-term cardiovascular disease after trastuzumab or anthracyclines, but have you looked at people who didn't receive chemotherapy but are getting hormone suppression, such as tamoxifen or exemestane or some kind of aromatase inhibitor?
Yeah, that's a really great question, and a study that we have currently ongoing in the works, and hopefully we can present that maybe next year.
Is there to build on that, your data on cardiovascular disease after treatment with chemotherapy, makes me wonder a little bit about whether there is a baseline increase even in the non-chemotherapy treated patients in cardiovascular disease compared with the normal or the non-cancer population.
That's a great point. I think one of the main aspects here is that cardiovascular disease and breast cancer are really similar diseases that have similar risk factors. Sorry, not similar diseases, but they have similar risk factors. So oftentimes, breast cancer survivors, by the time of their breast cancer diagnosis, are at an increased risk compared to women who are non-cancer. And there is evidence that women with breast cancer are more likely to develop and die of a cardiovascular disease compared to women who have never developed a cancer. So there's certainly that into play as well into this to consider.
Thanks. The SEER data that you showed, that's the SEER Medicare data, that tends to lag a couple of years behind. With the increase in use of immunotherapy, do you know if there's a signal there that might point in the same direction, or perhaps no comparison with chemotherapy?
I think with among the breast cancer population, the immunotherapy is more recent. And so to elucidate more of the long-term risk will take some time. And I agree, I think you're spot on with the SEER data seeing lag. But hopefully that's something that we can capture within the Kaiser Permanente cohort as we're continuing our follow-up. Our most recent follow-up goes to 2023. So we're really excited to be able to elucidate some of these questions in the future, potentially as we're capturing these detailed cancer treatments, including immunotherapy.
Thank you. I keep asking myself whether there's a fundamental immune reprogramming that happens in patients with breast cancer that may also apply to other disease states like cardiovascular disease, whether it's atherosclerosis or arrhythmia or perhaps autoimmune disease as well. So I think this is a really interesting area, and I think we'll stay tuned for what you present perhaps next year or soon after. Thank you, Dr. Vo.
Thank you.
We'll stay on the theme of immunotherapy. Dr. Rosa Wen is our next presenter from the DSP 2023 cohort, who will be speaking with us about immunotherapies for pediatric neuroblastoma from the bench to the bedside. Thanks, Dr. Wen. The floor is yours.
Oops, wrong one. Sorry. Okay, thank you, Dr. Kanti. I'm excited to be here and want to use my 10 minutes to talk with you about immunotherapies for pediatric neuroblastoma. And I'm going to start by showing a picture of my lab. We are actually the pediatric solid tumor and immunotherapy section, a very diverse group of people from 7 different nations, fun, as you can see, but also very dedicated to come to the lab every day to work on developing new immunotherapies for children with solid tumors with this focus on neuroblastoma. And we have different approaches of doing this, and I'm going to share with you one of these approaches today, which is integrating gamma-C cytokines to enhance different immunotherapies. And among this group of gamma-C cytokines, we are particularly interested in interleukin 15 and 21 to improve antibody therapy and CAR T-cell therapy. So neuroblastoma is a solid tumor that originates from the neural crest. Young children and toddlers are predominantly affected, and pediatric oncology or cancers in general are very rare, and among this group of patients, we actually only have 650 new cases a year, but that number puts us at the most common extracranial solid tumor in childhood. And about 50% of newly diagnosed children with this cancer are at high risk for relapse refractory disease or death. So it's not surprising when I tell you then that 15% of all cancer-related deaths in children are caused by neuroblastoma. But it's certainly not because we don't treat them. They actually get the kitchen sink. And despite that, less than 30%, particularly within this population of high-risk patients, can achieve long-term remissions. A big breakthrough in the field was actually the FDA approval of this anti-GD2 antibody, the first immunotherapy in solid tumor. And in this phase 3 trial by the Children's Oncology Group, children with neuroblastoma that have attained a state of minimal residual disease were randomized to either receive standard of care or this anti-GD2 antibody with cytokines. And 2 years out, there was a 20% survival benefit. So the trial was closed, and this became the standard of care. And antibody works. It's quite a fascinating therapy in my mind. It brings together effector cells of the immune system and the tumor cells by recognizing high levels of an antigen, in this case GD2, and then leads to the the killing of the tumor cells by these effector cells. And then you can modulate this reaction, for example, by adding cytokines that either enhance your effector cells or suppress your tumor cells. And in a more elegant way, you can actually take those cytokines and tether them to the antibody so that you only have local exposure of these very potent yet quite toxic molecules. And that's what we did. We created these immunocytokines by using an anti-GD2 antibody and tethering IL-15 or IL-21 to them and compared them to an existing immunocytokine with IL-2, which is another cytokine from this gamma-C family. And we tested this in vitro and vivo. I'm showing you here some data from an in vivo experiment. So animals, these were immunocompetent mice, were engrafted with neuroblastoma orthotopically in the periadrenal space and then randomized to receive one of these 4 treatments. And we then performed bioluminescence imaging, and you can see that at treatment start, all of these animals had tumor signal. But by day 30, the ones that received no treatment or this immunocytokine with IL-2 had substantially higher tumor burden than the ones treated with the novel immunocytokine that we made. And in a separate cohort, we let these animals age out until day 100, and there was a clear winner. So the ones that received the immunocytokine with IL-15 had much better survival, which we linked to an expansion of these good macrophages, which are the M1 tumor-associated macrophages with treatment, and a much lower level of immune-suppressive T regulatory cells in the animals that received these 2 new cytokines compared to the one with IL-2. So this was really the preclinical rationale for me to go into the clinic with this. So now we are in the process of translating this into a clinical study using the anti-GD2 antibody and IL-15 first separately, and looking for tolerability and a biologic signal. And then hopefully this will be the foundation to pave the way for the clinical translation of this immunocytokine with IL-15. I'm going to use the second half of this talk to talk a little bit about CAR T cells. So I'm sure you know that CAR T cells, chimeric antigen receptor T cells, have really transformed the treatment of patients with leukemia and lymphoma. But in solid tumors, our responses were not that great. As you can see here in this table that summarizes all the pediatric solid tumor CAR T cell trials that have been done thus far. And focusing on this blue column here, you can see that we achieved complete remissions really in only a few patients. And looking a little bit closer, these patients had very limited disease burden. So arguably, there are a lot of differences between leukemia and solid tumors, some that pertain to trafficking, so cells have a harder time getting into the tumor. We have much less of a repertoire of antigens that we can target, and the cells, the CAR T cells, if we make them and infuse them, when they get into the TME, they encounter a very hostile environment that really prohibits their function and persistence. So the question is, how can we overcome these challenges? And again, we turn to those cytokines that I mentioned, the IL-15 and 21. And this was work done by one of the postdocs in the lab, Dr. Riona Okada. We used GPC2 CAR T cells in this project. These cells, or the GPC2 target, was discovered by Mitchell Ho here at the NCI. He made a CAR and gave it to us, and we optimized it for clinical translation, but already thinking about the next generation of CARs to make this even more potent, particularly for patients, for example, with large tumor burden. But what Riona did was he used IL-15 and 21 and engineered a vector to tether these cytokines to the cell membrane, and then tested them functionally. In this assay, he uses 2 different neuroblastoma cell lines, IRMA5 and SKNFI, and it's an in vitro, so cell culture assay, where you add every 2 to 3 days tumor cells to CAR T cells and see how well do these CAR T cells perform with the consecutive tumor rechallenge. And as this graph increases here, basically that means that the tumors grow through and populate the well. And you see very nicely that over time, initially, I should say initially, the blue and the red line really run together, the blue representing the CARs alone and the red, the CARs armored with the IL-15 and 21. But after 2 to 3 re-challenges, these graphs really go apart and demonstrate that the armored CAR T cells perform much better in culture. He was also able to show this in vivo in an orthotopic xenograft model where we inject these ARMA5 cells into the periadrenal fat pad and let these animals, engraft this tumor to a large tumor burden and then treat them. And it's not surprising that with large tumors, the CARs alone, similar like in the patients, do not do any damage anymore because the tumor lines really just persist up here. But when the CARs are tethered with IL-15 and 21, now you can see that the tumor burden really decreases by week 3 and 4. So, with this effort, actually, we are now looking into understanding the mechanisms a little bit better in the tumor microenvironment, what IL-15 and IL-21 actually does or do to the immune cells in the TME. We also did some more engineering to limit the constitutive expression of IL-15 and IL-21 to make it safer. And as I mentioned before, we have optimized the GPC2 CAR for clinical translation, which will be hopefully open next year in the pediatric oncology branch with me as the PI. And this is the end of this talk. I'd like, again, thank my lab as well as all the collaborators and mentors within the POB, outside of the POB, the industry partners, and our funding sources, as well as the NIH DSP program. And last but not least, our patients and families. And this talk came at a very opportune time because this month is Childhood Cancer Awareness Month. So I'm happy to take any questions. Thank you.
Thank you, Dr. Wen. That was really fascinating work. We have a question, I think, on the left side of the auditorium.
Hi, Rosa.
Hey, how are you?
It's beautiful work.
Thank you.
So,
Can I ask you to kindly introduce yourself for the folks who are online?
Sure, I'm Luis Franco, DSP scholar from 2020, I think. So, around there.
We were the same class. So.
Yes, yeah, as Jürgen. So, 2 questions on the ADCC approach. With the IL-15 conjugates, is it clear that it's just the number of local M1 macrophages that matters as opposed to, say, local expansion or activation of the NK cells that come in?
So we did that and looked at it, and the NK cell numbers locally in this model were not expanded, at least at the time point when we did our single-cell RNA-seq and the flow cytometry. But rightfully so, IL-15 is the juice for NK cells and T cells. And, there are clinical studies that show when you infuse it as a free-floating agent, that you get massive, first, extravasation, but then massive expansion of NK cells or T cells. So I suspect that had we let them go longer, we would have seen that. Plus, this is one of the correlative studies in the clinical trial.
The other question is what's the relationship between these tumors and the blood-brain barrier? Like for the antibodies to get to enough cells of the tumor, do they need to cross the blood-brain barrier, or do they not need to cross the blood-brain barrier?
So neuroblastoma is actually a non-CNS tumor, so most of the tumor burden is peripheral, but the molecular weight of these molecules is too big to cross. So we would probably have to re-engineer it into a dye body or something smaller to make it go through.
Yeah, exactly. I was wondering about nanobodies, right? Single domain, camelid type antibodies, which supposedly are, have much better penetration of tissues like that.
Yeah, I think it's a great thought. We're playing around with this a little bit, not with the GD2 antibody, because that is highly expressed in nerves and in the CNS. So got to be a little bit careful there, I think, with toxicities, but with the GPC2 antibody, for example. So yeah, it's a great thought.
Okay.
Thank you, Luis. To build on that, I was also thinking of this from the standpoint of blood-brain barrier transmission, and wondered what types of cytotoxicity might you expect to see, even if if it's not getting into the blood, crossing the blood-brain barrier. What systemic toxicities might you expect from unrestrained IL-15, IL-21 signaling?
Yeah, that's a great question. I think, what we already know is that it can cause serious neurotoxicity in terms of pain. And the chimeric version of it, I think almost every child with the first infusion has this pain. So we give morphine and, try to titrate it up to get not beyond respiratory depression. But there are other formats of antibodies where they are, for example, the St. Jude antibody with mutations that don't engage the complement system that have made it better. For this combination, I think there will still be pain. So we're giving the dinutuximab at the FDA-approved level, but we are dose titrating slowly the IL-15 because I think the overlapping toxicity will be your inflammatory response. So I think there you got to be careful. It can cause basically hypotension, respiratory problems due to pulmonary edema and all of this. So a slow titration over time will be important. Plus, the way I designed the trial is not to give it as a bolus, but as a long-term infusion, which I think will make it easier as well in these children. Yeah, but they've been through a lot. They're truly warriors, and I think none of us in the room here would be able to, tolerate a tenth of this treatment.
Wonderful, truly inspiring work. Thank you very much, Dr. Nguyen.
Thank you.
Our next speaker is Dr. Diana Montero from NCI, who's going to be speaking with us about protein crystallography as a gateway for drug discovery. I think the symposium is planned really well. The topics transition beautifully.
Okay. This is the point that I should have, turned my computer on when I was still sitting down, clearly. So, all right. We have visual. Perfect. Okay, so I think this is going to be rather different from everything that we've heard so far. I'm also slightly blinded by these lights, so this is going to be interesting. I am a protein crystallographer. I actually started as a medicinal chemist, and I took a very big detour to protein crystallography. And coming to NCI means that I get to go back to my roots in drug discovery, which has been wonderful, and I get to interact with the biologists because I am truly not a biologist. So my group is technology-driven, is technique-driven, and we're bringing this, and I think if you all just tune out of my talk, just look at the last slide mostly, and think about kind of the technology that we're bringing in, because I would love to hear about, targets and things of importance from the people that really understand cancer from the point of view of organs and cells and all of that, which is something that is a much bigger scale than where I work. But I'm going to start by, our Pac-Man is going to be with us for most of this talk today. And as a crystallographer and having spent some time in dynamic crystallography and time resolved, I'm of the opinion that proteins are molecular machines and we should look at them as so. So for me and my group, we determine high-resolution structures of, in this case now, oncoproteins. It is my first rodeo in the realm of cancer, but all of the technology is translatable. And we also look at protein-ligand complexes. And this means that we determine mechanisms. So at the point of view of, atomic resolution mechanisms, we define these protein-ligand protein-protein interactions, which are extremely important for developing of drugs. And from that, we get structure-activity relationships, and we do all of this in as high throughput as we possibly can to accelerate everything and minimize sample consumption, because as it turns out, the more complex the proteins that you work with, the harder they are to make and the more precious they become, and we need to take that into account. And I have a soft spot for the dynamics, and I think that dynamics are overlooked, and I will show you A quick example of a project that we started back in March, where dynamics really clearly had an influence on how the small molecules that were being designed were effectively incorrect at the beginning. So small molecules, we just learned about immunotherapies. I'm on the small molecule ward. This is still the highest percentage of FDA-approved drugs, so there is so much happening in pushing immunotherapy and gene therapy and all of this, but small molecules still represent a really big part of it. And also from one of the projects I'm not going to touch on, but will be on the last slide, as like co-therapies to really improve the ability of drugs to work. So for us, making smarter drugs, a smarter design, we're trying to target the relevant biology. We're trying to have targeted therapies, have lower off-target effects, and these are all things that we can really try to modulate when we look at the small molecules and so we look at the structures, but also the dynamics. So basically, here's where we are. I've discovered an important oncogenic protein, and I want to understand what its molecular mechanism is, and I want to make a drug that efficiently targets that molecular mechanism. Or I already have a drug for a protein, but it doesn't work very well, so what can I do? So this is really where this structure activity and dynamic activity relationships come in for drug design. All right, so I moved to NCI in October last year. My lab became operational in about February, and I'll show you at the end when my people joined. And this was the first project that we took on. Our little Pac-Man is actually called PrEP. It's a prolyl endopeptidase. I was approached by someone from the chemical biology laboratory about this protein. And so who's our collaborator, was making PrEP inhibitors. It is a serine protease. They're actually targeting a cysteine in the active site rather than the serine, which is pretty smart. And it cleaves peptides. It is involved in the maturation and degradation of neuropeptides, so it's involved in diseases of the brain, effectively, and peptide hormones. And it is effectively somehow, no one fully understands this completely, but it is connected to neurodegenerative diseases. So they first started by taking what was a previous structure of PrEP and doing some computational docking. And this means that they take some molecules and they dock them into the structure site. And they came up with this. So this is the molecule that they were looking at. You can't really see it here very well, but I'll show you a different view in a second. It's covalently linked in the active site. So they built some new molecules that they thought would mimic this very well, and lo and behold, they didn't work as well as they expected. So we came in and we solved some structures. The fact that this, as you saw before, this structure is dynamic posed some issues to get good structures out, high-resolution structures out. So we took their compound and we solved the structure, and it looks very different, and I can show you the overlay. It's not the same molecule, but the orange molecule is based on the green molecule, and you can see that it sits in a completely different position. And if that wasn't enough, I give you the 90-degree view. It's truly, a completely different position of that warhead. So this is to say that, your computational docking is very useful, but if you have an active site that is huge, that it's dynamic, and you start with a static crystal structure and you design from that, you run into problems. So now we have these structures at hand, and now they are starting to redesign to try to make more potent inhibitors. So we are also looking into getting these structures at room temperature, so all of this is done at cryo temperatures, and that's also going to change. The active site is huge. So how are we going to expand on this? So it's a huge active site, it's highly mobile. Clearly the in silico is a problem. So even though they've already started, because we found some interactions that were suitable to guide the structural design, they're making new compounds, we are coming in with what we call fragment-based drug discovery. I'm going to take you, for those of you who know what this is, you can tune out right now. For those of you who don't, do stay with me, because I think it's pretty cool. Fragment-based drug discovery is the backbone of the vast majority of small molecule therapies that are out there, and industry has truly taken a hold of this for many years. From a technology perspective, it hasn't really been amenable to a lot of academic researchers, and effectively one of the first jobs that I had coming here was to establish that pipeline. So the way that it works is that this is a tray, it's a 96-well trays where we used to crystallize our proteins, and we effectively put these little 200 little drops of proteins, and there is a crystal that's a real crystal from PrEP. This is something that we collected data on. And then what we do is we take a fragment library, so these are very tiny molecules, and we soak them in. And we have effectively not just 96, but about 1,000 of them. And then this is the hard parts. We manually mount them, and there is no way around this. So my group, within the 5 people, they have been told that even if it's not your project, when we have this, you're going to sit down at a microscope and you're going to help look those crystals. And they're okay with that. So I think, I don't know, I hope too. So, lead by example every time. So when we have all of this, the other important part is that we do, so the initial data collection, when it's, just a few crystals and we're trying to just solve a structure, we do it manually. But eventually we automate this. And we have very close relationships with the facilities where we run this, and we're talking to them constantly to get this automation to work really well. So effectively, there's a robot. If anyone at any point wants to come and watch some data collection, it's pretty fun. Just come and let me know. They load, they shoot x-rays at it, they collect the data, they do the automated data processing. We get these thousands structures out, and then of course we're not going to look at them by hand. So there's some other software that was actually developed by a friend of mine who's now in Sweden called Panda that does filtering. And what we get on the outside is maybe 10% of hits, where we actually have something bound to the active site that is new, is novel, probably new chemistry, something that we haven't seen before. And then we do the manual inspection and correlate it to the library. So, of course, what I'm about to show you is purely theoretical, because as I said, we started in February, so this is our first campaign. We don't yet have data to show you, but the way that works is you have, in the case for PrEP, we have a huge active site. We're going to load the protein with a shorter version of these warheads that we know works, and we want to know is how we expand it. We load it up with fragments, and I don't know if you can see very well. I can't with the lights, but you basically get some other fragments in your active sites, and then you do some chemistry, and you're trying to link them to make a new molecule that should be tighter binding, more specific, all of that. So this is our pipeline. We typically start with protein expression purification. That is the biggest time that we spend is trying to make these proteins. We have a lot of infrastructure, so things that help us run in high throughput, so liquid handling robots, and we do this with very high accuracy, reproducibility, and very low sample consumption. We then go to the synchrotron. This is where we collect our data. This is a massive accelerator that produces very bright X-rays. So mostly do this remote now, but I try to go a couple of times a year there. Then we come in and we do, once we have some initial structures and we're ready to go through high throughput, we have an acoustic droplet generator, so this delivers the library. This is really important because crystals are very sensitive, so you don't want to touch them, so we do everything acoustically, and we have a setup that helps us mount the crystals. And really what we're doing is we're bringing this infrastructure in, so right now we are finalizing the instrumentation in the lab and the pipelines, and it is unique, and it is similar to one other institute that has it back in Cambridge that was the first one that brought this to academic science. So we have other projects. I'm not going to have time to talk about this, but we have projects related to this a quantitative carboxylase that is actually involved in metabolism, and it's been shown to be highly expressed in tumors when people have really bad outcomes with their therapy. So we're looking into potentially making some drugs towards this. This was the one project that I brought from my previous life, so antimicrobials against opportunistic pathogens, especially bad, 60% mortality rates. They come in if people are in long-term care. We have something called PROTACs. These are bidentate type molecules that not just bind to your protein, but they trigger the degradation, so effectively you take your protein out of the equation. And highlighted here, this is kind of my holy grail, which is we're trying to deal with oncogenic fusion proteins, and these are very difficult because, as you can see here, they're very spaghetti-like. So our first point is to try to do a lot of computation to find our approach currently has been to find binding partners and try to define ordered regions that we can look at and then disrupt with drugs. So with that, I want to say thank you to my group. As I said, most of the people working have started in April, May, so all of this work up to date has been done by them, and they've been really pushing it. And I hope you keep this in mind. So if you do have a target protein that you're interested in and making new drugs, do come and talk to us and make use of this technology that we have available. And thank you.
Thank you. That was fantastic. I think I want to come make a field trip because I want to see this in action.
Sure.
Do we have questions from the audience at all?
Hi, great talk. Jonah Nguyen, I'm actually with the 2024 DSP.
Hi.
I have a project. I'd like to collaborate with you.
Okay.
So maybe we can connect later. If that's
Sounds
okay.
great.
And that's all I want to say. Thanks.
Fantastic. That's what we're here for, right? So
You started with endopeptidase, and then you talked about carboxylases, and I wondered, there was on one of your final slides, there was you mentioned an oncotarget protein, but that didn't seem like an enzyme. So would you maybe give us some insight on what's the difference between trying to design molecules that will bind to active sites and enzymes versus non-enzymatic?
all right,
targets?
so active sites in enzymes are the typical targets, right? So they're pretty constrained. You have very clear interactions that happens between your protein and your substrate, so this is typically what you target. You do have a problem that if you're targeting something that has a substrate that is actually very similar to other things that happen in the cell, you might actually have off-target effects, right? And then the other problem with those is that typically you also start gaining resistance, so single point mutations in your active site and your molecule is dead. In terms of other things that are not enzymes, typically they work by, well, in the majority of cases that I know, they work by contacting with other proteins or other larger entities in the cell. So now we're looking at protein-protein interactions. It is known that these, so if we look here at this simulation, there is an area right around there where these things are contacting. They're much larger areas. They're kind of flat normally, and you do have some contacts, and it becomes a lot harder to target just from the sake of having enough contacts that make something that binds properly. But you suffer from less mutational burden that kind of kills your therapies, and you can do this and disrupt very specific interactions. For us, we probably will try a slightly different library of compounds. So there's libraries of compounds that work better to make hydrogen bonds, etc. Libraries of compounds that work better for surface-based things. So that's from this point, we can still do it by crystallography. We're just not going to be looking at an active site. We're going to be looking at a surface binder. But it works exactly the same way.
Quick question.
Hi.
Hi, I'm Flor Prato, 2023 cohort, same one, yeah. So the latest iterations of AlphaFold are promising to help with this. What do you think? Is it? Okay.
So that is AlphaFold. It helps, but just like anything that is I don't know if anyone has run AlphaFold. AlphaFold has a tendency to pick things that might not be real. So we use it, especially for things like the oncogenic fusion proteins where we're trying to see. So, okay, quick background. This thing that you see here, it actually started with a list of hits from bio-ID assays, so things that are close together in the cell. And now we're effectively going through 400 pairs and seeing if we can see any kind of indication that those interactions might be real. But we take it all with a pinch of salt. So if we find anything that looks like this, that has like reasonable contacts, we are going to try and go after it. We're going to have to make the fragments. We're going to have to see if they actually bind, and if they do, then we follow through with a drug discovery assay.
Still a hypothesis generator.
It's completely a hypothesis. If anyone gives anybody an AlphaFold model and tells you that it's real, they're very useful, but you kind of cannot take it as point value, and we've seen that. So, yeah.
Wonderful. Thank you very much, Montero. This has been really wonderful start. We're going to take a 6-minute break and resume at 11:15. Thank you. Thank you, Bryce. Saving the day. The structure of the symposium today is that we're going to have 3 different groups of DSP scholars presenting their work. And so the second half of the first cohort is what we'll meet to hear from in 5 minutes. Thanks. Hey, good morning. Welcome back. Looks like we've all come back. Everyone got a chance to stretch your legs. We don't want anyone getting a venous thrombosis, which is what I study. So flex your ankles a bit and make sure you got some blood circulation. And so really excited about our next speaker. This is Dr. Min-Kyo Song, who is joining us from NIH and will be speaking with us about the immunoepidemiology of autoimmunity and aging.
Thanks for the introduction. My name is Min-Kyo Song. I'm a tenure-track investigator at the National Institute on Aging. I'm leading the immunoepidemiology unit within the Laboratory of Epidemiology and Population Sciences. I'd like to thank the DSP for supporting my research, and I am very excited here today to talk about immunoepidemiology of autoimmunity and aging. Immunoepidemiology is a combination of 2 disciplines, immunology and epidemiology. And it can be defined as a field of study that addresses how differences in immune responses among individuals affect the epidemiology of infectious diseases, cancer, hypersensitivity, and autoimmunity. As you can see, immunity affects life in various ways in aging. When a person gets born, the immune matures with the aging process, and eventually immunity also gets old, i.e. immune senescence. In the immune senescence stage, thymus regresses and the number of naive T cells go down, vaccine responsiveness reduce. On the other hand, the infection rate, cancer, and autoimmune diseases go up. In order to answer this overarching research question of what is immunity's role in aging and age-related diseases, one approach is to look at altered immunity, and amongst which I am specifically looking in autoimmunity. And you can see on the science cover in 2022, I don't know if it's probably too small to see, but autoimmunity underneath that says is the dark side of the host defense, meaning immunity that's supposed to protect the host goes wrong and is attacking itself. That's the definition of autoimmunity in short. I'd like to say this is an exciting and timely opportunity to conduct research in autoimmune disease on aging. Number one is that it's an increasing burden of autoimmune diseases. It's estimated that around 10% of the world population is affected with autoimmune disease. But the bad thing is that there is a 19% net increase in incidence per year. And autoimmune disease patients are living longer thanks to the treatments. But another thing is that the women are more affected by the disease, meaning that these are the main caretakers of the family, and in the downstream will affect the household and also the community much more. There's still limited understanding of autoimmune disease epidemiology. We don't know the demographic, temporal, or geographical variations of many of the autoimmune diseases. And we lack lifespan or longitudinal designs, specifically in the epi designs. And not many of the studies take into consideration of these emerging risk factors, such as new drugs, infections, or climate change. But we are in good hands. There's advances in immunology and statistics, and there are more data available out there. Not just the classic epi design studies, but there are secondary studies that we can utilize. And the National Academies of Science and Engineering, they thought that this was also a great need. So they produced this consensus study report in 2022 for enhancing NIH research on autoimmune diseases. And in response to Congress, the Office of Autoimmune Disease Research was created within the Office of Research on Women's Health. And then more recently, the White House Initiative on Women's Health Research, they also emphasized autoimmune disease is one of their priority areas. In order to answer what is autoimmune's role in aging and age-related diseases, one of the biggest challenges is to disentangle the directionality of aging and autoimmunity. As I said, with aging, autoimmune diseases increase, but also it is true that with autoimmune diseases, age-related disease such as cardiovascular disease, Alzheimer's disease, and cancers also increase. And we're not so sure about this association in different risk groups. So there are two different aims of my research. One area is to unravel the association between autoimmune and aging. In order to do that, I'm looking at the effect of aging on the risk of autoimmune diseases and also the effect of autoimmune diseases on the aging process. In order to do this, I'm incorporating longitudinal study designs, lifetime course, and also these new risk factors such as infection, medications, or climate change. The second area is to develop methods to better ascertain autoimmunity. Autoimmune diseases are often underdiagnosed, and a lot of autoimmune disease patients are asymptomatic. So what you see in the field of autoimmune diseases is just the tip of iceberg. So in order to better quantify autoimmune diseases and also autoimmunity, I am utilizing the clinical biomarkers and also developing new biomarkers for detecting these autoimmune diseases. Today, I would like to just introduce you to my 3 exciting projects that I've launched since onboarding last year, which would involve the longitudinal study designs and the life course designs, and also quantification of autoimmune diseases, and also incorporation of climate change. So I did tell you that autoimmunity increases with age, but there are not so many data out there that has documented the autoantibody change, the levels of autoimmunity change within an individual, or whether these trajectories differ by sex. So I am utilizing this study that's called Baltimore Longitudinal Study of Aging, which is a unique study that is America's longest study of aging since 1958. That has more than 3,000 volunteers with longitudinal data, with lots of surveys, various clinical and biomedical measurements, functional measurements, and biospecimen. I am incorporating the case cohort study where I'm selecting 250 autoimmune diseases and comparing that to the subcohort of 200 randomly selected individuals. And then I'm measuring the anti-nuclear antibodies, which is one of the most common autoantibodies that are found in many of the autoimmune diseases, and also am incorporating the novel technology that's called nucleic acid programmable protein array, in short, NAPA. And I'll be selecting 300 autoantibodies to see whether there are any autoantibodies that are documentable within this population. So I'll be looking at age-related changes of these autoantibodies and also these autoantibodies' relationship to the autoimmune diseases and other types of age-related diseases other than autoimmune disease. I have a lot of collaborators there, and this has been co-funded by the Office of Autoimmune Disease Research I mentioned earlier in the presentation. Although BLSA is a unique and very precious data set, it's pretty small to an epidemiologist like me, and since autoimmune diseases are rare in the population, I needed a bigger data set. So I went to seek for this large data set, which is Hispanic Community Health Studies, City of Latinos, that is an NIH-funded cohort that encompasses 16,000 Hispanics with a wide age range that covers the younger years that are more prone to age-related autoimmune diseases. It has a rich data collection, biological lifestyle factors, along with the social determinants, neighborhood, environmental factors. As you can see on the figure on the right, there are just so many things that you can utilize there. So here I'm also defining autoimmunity using autoantibodies. I'll be selecting autoantigens from the AAG Atlas, which is a literature-based accumulation of different autoantibodies. And then I'll be testing around 1,700 autoantibodies within the 10% of the population to select the top most important, which is going to be around 300 autoantibodies within the 16,000 populations. And then I'll be comparing those autoantibody levels with various risk factors that are surveyed or measured within this cohort. And also, I'll be looking at their associations with infectious agents, because infection is supposed to be one of those triggers for autoimmune disease. And then I'll be able to look at their associations with later risk of age-related phenotypes. And then one important thing is that since the cohort is distributed across different areas of the U.S., they have GIS measurements, and there's a working group for climate change, and I plan to collaborate with this group to look at the climate change that I'm interested in. And that's a nice segue to my last project that I'm going to present today, is the climate change consequences on the markers of senescence. So amongst many climate changes, I've looked at hurricane, and hurricane is supposed to be have indirect or direct effects on human health through socio-psycho-environmental stress on the acute and intermediate effects, and then finally to the chronic or structural effects. I'm utilizing the action to control cardiovascular risk in diabetes Accord study, which is an intervention trial of intensive glycemic control of 10,000 people with longitudinal specimens. This is a unique study because it has been affected by Hurricane Katrina, especially the southeast network, as you can see on the map. And so I'm comparing the Tulane study, which is located within the Louisiana, colored in red, and comparing them to the other clinics colored in yellow, and using the difference in difference method, try to figure out the additional effect of hurricane on these biomarkers. I'm using Olink, which is one of the most famous proteomics platform, and we'll look at the changes of protein signatures pre and post Hurricane Katrina, and also to find out which of the protein markers are predictive of these differences, and the differences of cognitive function, which is included in our study. And this was awarded and funded by the Intramural Targeted Climate Change and Health Program 2023. I'll just go short. Take-home message is that immunoepidemiology integrates the principles of immunology onto the field of population science. Autoimmunity is one approach to answering immunity's role in aging. There is high demand and interest and prospects in conducting research in autoimmunity. And in accordance with the physiological change in the individual's immunity, longitudinal assessments of autoimmunity is critical, and that developing new biomarkers of autoimmunity may help us understand the aging process, and hopefully the identification of high-risk individuals may provide opportunities for intervention at early stage. And I can't do this by myself, and here's my team that is supporting me. Okay, thank you.
Thank you, Dr. Song. That sounds like very, very interesting and important work. We have time for one question. Okay, great. I love it. I'll ask the question. It's great. It's good to moderate because I can use the time how I want. So I keep looking at this data of the increased incidence of cardiovascular disease, cancer, neurodegenerative disease, autoimmunity with aging, and I'm asking myself, is there anything good about getting old?
Physiologically, you mean?
Mm-hmm.
I don't know. I haven't really given that thought. But one part is that you're getting wiser and you're getting more tolerable, and you don't get so much stress in sessions like these.
Okay, wonderful. We'll leave it at that then. Thank you very much, Dr. Song.
Thank you.
Our next speaker is Dr. Manu Platt from NIBIB, who's going to be speaking to us about the dynamic interplay between biomechanics and biochemistry and, guess what, a vascular disease, sickle cell large artery damage.
Let's do it. Okay. All right. Is there a laser pointer here? This is one. I can use one of these. I'll use one of those. This is one of these. Oh, I can use my cursor. Yeah, let me do this. Okay. All right. Great. Thank you all for having me. It's great to hear from my other distinguished scholars, and especially those in my small mentoring group with Susan Buchanan. So I found out just from Mike got us at the break, I'm a senior distinguished scholar, which, good things about aging. So I'm in the National Institute of Biomedical Imaging and Bioengineering, and I'll be talking to you about my work regarding sickle cell disease. But not only was I recruited to be an intramural investigator, but also to be the director of the newly formed Beta Center, or the Center for Biomedical Engineering Technology Acceleration. And my wonderful deputy director, Nicole Morgan, is here as well. And so our goal is really, I think, to help bring biomedical engineering principles to the NIH IRP and to help other investigators, clinicians, to access some of these technologies and build community here on campus around biomedical engineering, as well as using diversity, equity, inclusion as some driving principles for what we do. And so with that, we want to offer our service of science concierge service to you all. We have been partnering, of course, with the SRS resources that NIBIB already manages that you all can work with, but bringing online some new capabilities such as systems and computational biology, biomaterials, or soft biomaterials, and we are building NIH's first makerspace for you to come in and modify and make your own tools and research for your activities coming online soon. And you can see we built up our staff. Coming up, Castle Kim is our makerspace manager, and Jacob and Carrie are two of our staff researchers that will help get your work done. So there's our email address. If you're not affiliated in Beta Center and want to be on our listserv and get access to our things, please send us an email, and we're happy to add you. We have lots of activities, meet and greets to get us to help know each other on campus. But some of the other, we also run training programs, again, to recruit new bioengineers to campus, help show them what the NIH IRP actually can do, because a lot of them out in the world don't realize about the exciting things happening here. And so one of our programs that now comes under Beta Center is the long-running BSIP program, Biomedical Engineering Summer Internship Program. And for those that don't know, NIBIB funds these students, but we distribute them to investigators across the different ICs. So I'd love to see more distinguished scholars submit projects for this upcoming summer, and then you can mentor one of these students right now on NIBIB's cost. But if other ICs would like to contribute funds so we could expand the BSIP program, we are very excited to have you join us. And we've opened it up to not just biomedical engineering majors, but also STEM majors that are interested in maybe pursuing biomedical engineering in the later parts of their careers. So they're fantastic students. Also, again, another opportunity is October 22nd. We are hosting a big bioengineering day here on the NIH campus. If you haven't heard, I'm pitching it. You can still apply, free registration, of course, where, again, we're trying to bridge extramural and intramural. So we have the Biomedical Engineering Society annual conference is in Baltimore this year. So the day before, we're having everyone here to Bethesda to come and meet us, a blend of talks, posters, intramural, extramural, talking, networking, and kind of pitching our wares to them and getting them to want to work with us. 2 keynotes, one from John Tisdale, an intramural clinician who's been groundbreaking on gene therapy for sickle cell, and Rashid Bashir, who's a leader in the outside world, does amazing things with diagnostics. Okay, now I get to talk about me. Again, wearing those 2 hats, I always get my time cut short because I got to plug my Beta Center work. But I do run a lab here. I'm thankful to do that as well. I run the Matrices Lab, mechanics and tissue remodeling, integrating computational and experimental systems. We do a number of different things, and I hope to show you a little bit about my work to show you how engineering principles can be used to ask biological and clinical questions. But I wanted to start, I saw Diana's talk. She's in my mentoring group. I didn't know she did proteases. I didn't know she was a crystallographer. But I like to promote myself. I love proteases, okay? More than anything. This is me at the GRC on proteolytic enzymes and their inhibitors, and I am advertising for NIH. And my postdoc, Jacob Davis, was with us also. But I think about the context of sickle cell disease. Then in the context of sickle cell disease. And September is Sickle Cell Awareness Month, so I heard that it was also another month, so shout out to that. But there's a single point mutation we've all heard about, but it causes multifactorial damage, and life expectancy is still lower than general population. Homozygous recessive, so SS, has sickle cell, and it's a health disparity in the United States. Predominantly affects African Americans. 1 in 400 are SS, 1 in 12 have sickle cell trait or are carriers. And it's estimated about 100,000 Americans with sickle cell, but it's a global disease. But what really got me into it was when I found out the cardiovascular components, because you know the red cells sickle under deoxygenated conditions, so a lot of work in the microcirculation. But when I started my independent research group, I was told that children with sickle cell were having strokes. What? My PhD training was about cardiovascular disease and heart attacks and strokes that develop over 60, 70, 80 years. These are children having strokes, which doesn't make sense. Strokes, again, are diseases of the oxygenated conduits, and I was told that the cells sickle under deoxygenated conditions. None of it was making sense, but this phenomenon was happening. So 11% of children with sickle cell have a stroke by age 16. The greatest risk between the ages of 2 and 6 years old, so you can imagine it pulls on the heartstrings. But up to a third will have what we call a silent stroke, which is in a cognitive part where it doesn't present until they look at an MRI and they realize the challenges. So how does it come with engineering? Well, there was this amazing clinical trial called the STOP trial done in the early 90s that showed that they did transcranial Doppler. Children with increased centerline velocities were those at greatest risk of developing strokes. And they put them on monthly blood transfusion protocols, which completely was revolutionary. One of those clinical trials that stopped early because those that were not receiving the transfusions were having strokes or dying. So this is still current standard of care. So the question was, well, why are the velocities increased? Well, I learned in my fluid mechanics class that where you get stenoses or closing of the artery of the lumen, that you can get increased velocities through that area. But what we also know is that post-stenoses, you get these regions of disturbed flow in the vasculature. And we know that where those regions of disturbed flow happen, those are sites where atherosclerotic plaques were forming. So this was from the 60s. You can see a pathologist started to see atherosclerotic plaques at these areas of bifurcations and sharp curves. Talked to his aerospace engineer buddy who said, oh yeah, well we know with flow, if you have a bifurcation, you get these regions of flow recirculation and flow reversal over the cardiac cycle. And where you get those regions is where we're seeing the atherosclerotic plaques forming, which leads to just the mechanical forces of blood flow increase or influence gene and protein expression for health or disease. So I was like, wait, why are we not looking at this in sickle cell disease? The importance of diversity in sciences. We all bring our perspectives to a problem. Things we think others are looking at, no, your own perspective wasn't critical. So we began to partner with clinicians who sent us some magnetic resonance angiogram scans from patients that we could recreate in the computer. So then we could use computational fluid dynamics to look at how the geometry and the anatomies of the cerebral vasculature could lead to regions of flow recirculation and disturbed flow that might be susceptible to this accelerated vascular remodeling in the arterial branches of children with sickle cell. And we saw that only SS children were reconstructed had actually these regions of flow recirculation. And this was in humans. And now, as an engineer, we can recreate these different fluid profiles in vitro, or we can have a cone and plate bioreactor. Imagine a monolayer of endothelial cells cultured on the bottom of that dish. And now you can put in vasoprotective profiles, or those at the straight parts of the arterial tree, or a pro-remodeling profile like those seen on the outside of the carotid artery wall that induce vascular remodeling protocols. And the endothelial cells, they elongate and align in the direction of flow. That's how you know they're working and your shear system works. And you can also look, this is where there's this interplay of the biomechanics and the biochemistry. Lots of inflammation in sickle cell, but there's complicated interferences between the different flow profiles that are turned up and the biochemistry responses. So that's what we do with humans and human cells, but of course we have mouse models. There's a sickle transgenic mouse model, knocked out the mouse hemoglobins, knocked in the human hemoglobins, so now you can breed it with Mendelian genetics. And one of our first things we saw, we were looking in young mice because we were thinking about pediatric strokes with sickle cell. And cathepsin K, one of my enzymes that we showed was activated under disturbed flow, is elevated in the carotid arteries of mice with sickle cell. But through other work, we found that inhibiting that J and K C-Jun signaling axis can actually stop cathepsin K from being turned up, even in mice treated with that inhibitor. But we then looked at later time periods. So these are now 5-month-old mice. And this really shocked us. We were really focused on elastic lamina degradation. But by 5 months, we saw that this incredible loss of type 1 collagen in the artery wall. Collagen conferring stiffness to the artery wall. And you see this blown out aneurysmal artery. And it's known that once the children get past the risk of stroke, when they become early 20s, mid-20s or 30s, they're at risk of hemorrhagic strokes. So then this loss of this mechanical integrity to the artery wall could predispose them to that. And now we've been looking at that. But there's a lot of variability. I told you 11% of children have a major stroke, a third will have a cognitive stroke. The same variability is passed on into our mouse models. We love a good clonogenic mouse, but whatever. And so my senior staff, or my staff scientist, identified an MRA protocol that we can longitudinally monitor the same mouse as they age to reduce that subject-to-subject variability. And so we've been doing that, and over time, from 1 to 5 months, monitoring the changes in the diameters of the arteries of these SS mice, the heterozygotes, and then their wild types, and then averaging that luminal area over time. And so what we were able to see is that, yes, with living mice, we are able to see this luminal expansion as the mice age, which matches what we were showing before. But then the question is, how does that not change the biomechanics or the blood flow profiles? So here I've got some talented postbacks who've been doing CFD. And again, along the length of that artery, the fluid profiles are changing. I hear you. And now we can recreate that and look at what is the distribution in the SS arteries versus the others. And what's really interesting is you see these greater areas of high and low shear stress in the SS compared to the two arteries and in the straight parts of the artery wall. And that becomes the distinction between cardiovascular. disease in the others. And now we're trying to register where we see those fluid profiles with the actual biological changes that we can look at with histology and immunohistochemistry. And then finally, HSCT is the only functional cure. There's the gene therapy that's worked, but it will still require bone marrow transplant. So they're showing that even patients with transplants still are at risk of having strokes later. So we want to understand if there's adjuvant therapies that we could do, or if the timing of the HSCT could be helpful. Just to breeze through it, we did transplants either 2-month or 4-month, and definitely yes, the earlier we do the transplant, we could protect the vasculature from accumulating the insults, and that works out for the mice later. So anatomical changes are important. Think about the mechanics of blood flow as well as the biology, and the patient-to-patient variability means we might also have to consider not only the curative transplants, but any adjuvant therapies to protect the vasculature afterwards if that's not sufficient. So thank you. Love my team. Many are here in the audience. I want to thank the DSP program, Dr. Marie Bernard and Jean Shin, really supportive to help pull me here. And I got to thank my director, Bruce Stromberg, who influenced me and persuaded me of the wonders of doing work at NIH. And it's been really wonderful being here. So thank you all, and come to Beta Center.
Wonderful. Thank you, Dr. Platt. That was a tour de force, and thanks for the great advertising for the Beta Center. You know, I've heard a lot about this, and I think we've got to connect at some point soon. Wonderful. Are there any questions, Dr. Platt?
Yes. Thank you for the wonderful talk. So I'm an endocrinologist, and I'm trying to figure out if some of the work that you do, specifically looking at blood flow, is applicable to cohorts with diabetes, where we see a lot of microvascular complications. So if some of your work, I know you focus on a lot of the bigger blood vessels, but if principles of some of what you do can also apply to smaller blood vessels and microvascular complications in diabetes.
Absolutely. And so, absolutely. And so we do MRAs on the mice for the large vessels, but we can also euthanize them and do micro-CT, and that gives us all the volumes and the anatomies of the microvessels, and we can also use those in our computational models. I'm happy to talk to you afterwards, for sure.
So you're open to studying people with diabetes in addition to sickle cell?
Oh, for sure. Yeah. Well, we're studying the tubes. The tubes are the tubes. Yes. And Beta Center can help you and your team get started with all of those things as well.
Thank you.
Yeah, thank you.
Wonderful. I cut my teeth as a postdoc doing these cone plate viscometers in the same lab where you did your studies. So, okay. So I see some parallels there. I wonder, when we think about blood vessels, we think about them mostly in terms of the flow that's going through in one direction. But you described that there's a stiffness. So there's, not only is there a radial, there's an axial stretch that goes along with this. Do you think that there are ways to potentially reverse this phenotype once it sets in? This is getting back to the aging thing. As you get older, your blood vessels get stiffer.
So I didn't show this because 10 minutes are short, but we also have mechanical tester devices in our lab that do arterial stiffness and even the in vivo axial length. I think you're the vascular person. I think the field still says we cannot correct damage to the arteries. We know for sure we cannot generate new elastin that is cross-linked, that provides the elasticity that the native elastin did. I think if you can maybe modulate some of the collagen so it's not too stiff, but the risk there is if you reduce it too much, and then you're at risk of blowing it out and having a hemorrhage. So I don't think yet, but I think there's regenerative engineering principles where people are trying to find new ways, but I think the native body doesn't repair on its own.
Thanks. And sickle cell disease being a hemoglobinopathy, do you think these principles apply to some of the other hemoglobinopathies, whether it's some of the thalassemia?
Yeah, so we do think that there's some of these similar things that are happening in the alpha thal and the beta thal, but we're focusing on the sickle cell first. But we focus on SS sickle cell, but we're now also starting to get into some SC. There's been some interesting things that have come up with that later. And to the endocrinologist, I was hoping you were going to say osteoporosis, but you didn't. But that's fine, because there's also sickle bone disease complications that my lab is also looking at. So it's a multifactorial, affects multi-organ systems, and yeah.
Wonderful. Thank you very much, Dr. Platt.
Appreciate you.
Our next speaker before the break is Dr. Flora Prato from NIDDK, who's going to be speaking with us about multi-layered control of recombination and diversity in the mammalian germline.
Thank you. Hi, thanks a lot for being here. Glad to be part of the cohort. And yeah, perfect. So I'm going to, tough act to follow, Manu, but I'm going to try my best. I'm going to tell you something different about the multiple layers of control of recombination and diversity in the mammalian germline. So my lab is interested in studying meiosis. Meiosis is the defining event of mammalian gametogenesis, and it is necessary to generate haploid gametes from a diploid cell. And it does so by appending 2 rounds of division to a single round of DNA replication. And we're mostly interested in the events, let me click here, in the events that happen during prophase 1 and the hallmark event of meiosis, which is meiotic recombination. It is initiated by the deliberate introduction of double-strand breaks, hundreds per cell, and they are then resolved as a crossover. So crossovers are this allelic exchange between the chromosomes, the homolog chromosomes, and it serves 2 main purposes during meiosis. One goal of meiotic recombination is to physically link these pairs of homologous chromosomes so they can properly orient on the metaphase plate and segregate properly. And second, it creates new allelic combinations of paternal and maternal alleles. Therefore, it creates genetic diversity. So these double-strand breaks are very important for successful meiosis, but they are dangerous lesions. This is a silly game to play. And the cell has to make sure that they are made in the right amounts, at the right time, in the right places. So even though there's a lot of good control of these events, meiosis is a very error-prone process, particularly in human females. And it's been estimated that up to 60% of human eggs actually have a chromosomal abnormality. Therefore, the errors in meiotic chromosome segregation due to faulty recombination, many of these cases, are a leading cause of mental disability, miscarriages, and infertility. So we have spent a lot of time trying to understand how these events are made and how we control the outcomes. And so these are made by a conserved protein that's called SPO11, that is the actual protein that makes the double-strand break. Then there's a step of resection. There's a nucleolytic resection, a 3' overhang is exposed, and this protein called DMC1, that is a meiotic-specific recombinase, just like RAD51 or Recadin bacteria, covers the filament and then proceeds to do the homology search. So in meiosis, the repair is biased towards the homolog, not the sister. So once the homolog is engaged, these breaks are repaired, and in mammalian meiosis, most of the time they're repaired as a non-crossover. That is a short, it is an outcome of repair that doesn't does the haplotype switch, but leaves a very short gene conversion. So these programmed double-strand breaks do not occur at random. They occur at hotspots, so preferred regions of the genome where these guys are located. And back in 2010, it was discovered that PRDM9 is a very interesting protein. It's a major determinant of the hotspot location. It's a histone methyltransferase, a meiotic-specific histone methyltransferase that has a hypervariable zinc finger array. And through the variability on the zinc finger array, it binds to different places on the genome and therefore changes the recombination landscape among individuals. It is the fastest evolving gene segment in mammals, and we have found hundreds of alleles of PRDM9 from humans and mice. So they occur at hotspots, but we didn't know before we started this work where they were located in mammals. So we developed a technique that is a variant of ChIP-seq, in which we pull down the MC1 from this intermediate, but then we directly sequence the single-stranded DNA that's associated with it, so we keep the strand information. So we have a very stereotypical pattern of top strand to the left of the break and bottom strand to the right of the break. This affords us, gives us a lot of sensitivity and specificity, so we were able to do this from samples from human individuals. So we did that, and what we see is that this is a snapshot of meiotic double-strand break hotspots from human individuals. So you see that the positions of the hotspots are determined by the type of PRDM9 allele that this individual carries. But very interestingly, when we looked at the whole chromosomes, we see that independently of the PRDM9 allele, so this is the same for every individual, there's a huge increase of recombination initiation at the ends of the chromosome. So this is about the 10 megabases distal part of the chromosomes, and there's more hotspots and they are more frequent. So this past work uncovered 2 different processes that operate at fine and broad scales. One that is fast evolving through the fast evolution of the zinc fingers of PRDM9, and a second that is conserved through evolution, as illustrated here by the conservation of the subtelomeric bias on recombination from humans and chimpanzees. So we thought to understand what's underlying this broad-scale control of recombination, and we thought of looking into DNA replication just before. It's the main process that happens just before breaks are formed, and also in yeast people had linked recombination and replication mechanistically. But the problem, and I had to remind you that we cannot study mammalian meiosis in vivo. There's the in vitro, sorry, it has to be studied in vivo because there's not in vitro system. This is happening in the case of males in the testis. It's a complex tissue, and just about 1% of the cells at a given time are actually undergoing meiotic replication. So this posed many challenges, so I had to develop a method. And we came up with this tripartite method to characterize replication in mammalian meiosis, in which we first map origins of replication, just improving upon existing methods by coupling our single-strand DNA sequencing method to the origin detection. We then isolated replicating meiotic nuclei with high purity from using a combination of nuclei sorting. And finally, we integrated these two experimental readouts in a model of DNA replications to parameterize replication and understand what's going on in meiosis. So what we learned is that mouse meiotic replication is slower than that of the cells, other cells in the testis, like spermatogonia, but also in any other cell type that we looked. This was not too surprising because everywhere people had looked, meiotic replication is slower and takes about twice the time in yeast, for instance, or in newts or in lilies. People have looked at these organisms before. But what we could learn from our model is that this slow replication is due to lower firing rate. So there's fewer origins fired when cells do meiotic replication than in other cell types. So we're trying to understand why is that, how, and what's the consequences of having a slightly faster replication in meiosis. We went back to recombination, and this is from mouse, and we looked at any metric of recombination that we had in the lab, and we can find that PRD9 binding, double-stranded hotspots, and crossovers are all highly enriched in early replicating parts of the genome, therefore correlating these two processes. But very satisfyingly, when we looked at the human meiotic replication timing profiles, we see that there is a very distinct pattern, and it's germline specific, where we see early replication at the ends of the chromosomes. So this highly distinct pattern mirrors the distribution of the broad-scale distribution of double-strand breaks, therefore suggesting that this early replication is underlying the patterns that we see in terms of breaks and crossovers. So since then, we have applied this tripartite method to study replication to human testes. And we had detected origins and made this model. So we have now a very nice, finely resolved replication timing profiles from human meiotic. So I told you that there's a broad correlation between breaks and early replication and also crossovers. But now, with this very fine resolution, we can start asking different questions. And now when we look at the subtelomeric regions of the genome or the interstitial parts of the genome separately, we see a bit of a different story. So in the distal parts of the genome, there's more double-strand breaks and more crossovers at the early replicating parts of the genome, but that correlation breaks down in the interstitial part of the genome. So there's still breaks, but they do not become a crossover. So this suggests that there's the chromosomal context actually modifies the pathway that the cells uses to resolve these double-strand breaks. And this is very important because when we have hundreds of breaks and only 10%, I told you, resolve as a crossover, and how and where they are determined, it's a fundamental question still unsolved in meiosis. So the system allows us to start understanding the things. And just to finish, and I didn't tell you this, that in humans, remember I told you there's a female meiosis is quite error-prone, and interestingly, is hugely sex dimorphic. So male crossovers are enriched at the ends, and so are double-strand breaks, and we know now that these regions replicate very early in meiosis, but female crossovers are actually depleted at the ends. And one of the major risks for non-disjunction, for instance, for chromosome 21, and therefore Down syndrome, is having a crossover too distally in the chromosomes. So this system now is allowing us to try to understand how these processes are controlled, and I hope to have more questions in a little while. More answers, sorry, questions I still have them. And so just to finish, thanks the current members of the lab, Christina and Neshma, and my previous lab at NIDK, Duncan Marinatera lab, and all the collaborators past and present, and of course the Distinguished Scholar Program. Thank you.
Thank you very much, Dr. Prado. That's a fascinating talk. Are there,
How are you making this about cardiovascular?
I'm not.
Come on.
I have a way.
Yes, this is Rosa again. I thought it was also a fascinating talk. I have a question about the PRDM9 regions. So in childhood cancer, there are recurrent fusions and, that precisely recur in different patients. Mm-hmm. So are these regions that lead to double-strand breakage also associated with oncogenes and proximity, and do they contribute to that pathogenesis?
So, yeah, a lot of people have been looking into that because one of the, there's this field of cancer testis antigens, as they call them, that is meiotic genes that actually are expressed and regulated in cancers. PRDM9 is one of them. And there was one single study where they looked at children leukemia, and they had excess of some of the alleles of PRDM9. So there might be a correlation. So PRDM9 actually opens the chromatin because that's remethylation. If it's overexpressed outside meiosis, that could start opening the chromatin, and then you don't have SPO11 maybe, and so you don't have the whole targeting machinery, but it could definitely make an impact. There's not, so we know exactly where these guys are, and if you look, we have looked at different fragile replication sites and oncogenes and fusions, and we don't see the exact same position, but in cancers they are overexpressed and regulated. So.
I love that your talk started with more foundational physiological principles, and then you moved into disease states. And what I'm wondering about is, during meiosis, is the segregation of chromosomes random, or is there some design by which we end up with hotspots that segregate together?
Yeah, no. So that you have the major assortment of alleles is due to the independent assortment of chromosomes. But then with the breaks, what you do is, with the crossovers, you do the new allele generation. So it's known in females that the recombinant chromosome tends to go to it tends to stay in the oocytes instead of being extruded to the polar body. So there's something that's sensing that the recombinant you better keep the recombinant allele. So you have 4 chromatids, just one has the recombination event. Most of the time you don't inherit the recombinant, but if there's a small bias in female meiosis, particularly for the recombinant.
Really interesting. Is there a thought on what that mechanism might be?
No, it has to be something about the proteins that mark the crossovers. Yeah. Because other than that, yeah.
Luis?
It's a great talk. Why do you think such an error-prone mechanism would have survived evolution, right? The classic genetics textbooks will say that it's through the solution to Muller's ratchet of, you keep accumulating variants, and at some point you need something to either cluster them, the bad ones or the good ones together. I'm not sure that explanation is sort of fully satisfactory, but you're an expert. What do you think?
So mouse, for instance, they are not error-prone at all. So female meiosis in mouse is pretty good. Female horses are just like humans. There's many organisms that had this error rate very high, but not all. What people had proposed is that you don't want to have too many kids, kind of one after the other. So that's one of the ideas why it had been kept. Maybe we're at the evolutionary time where this is a glitch and then it's going to be fixed. Of course, that's not an answer I'm going to get. But so all sites kind of go for quality over quantity. So whatever is not working, so it's a huge attrition of oocytes actually female meiosis happens in the fetal ovary, and about 10% of the oocytes actually survive to birth. So there's a lot of pruning done there. So.
Great. Thank you. Great work.
Wonderful. Thank you very much, Dr. Prado. This is to err as human.
I like that. Can I use it?
Please do.
Thank you.
Well, thank you. Thank you. We'll close the morning session here and break for lunch. I'll remind you that there are posters still on the terrace until 12:30. So if you've got a few moments, please gander over there and take a look and provide some support to our trainees who are presenting their research here. Otherwise, we'll see you back here at 1 o'clock. Take a lunch break, stretch your legs, no DVTs, anyone, please, and then we'll see you back. Thank you.